In den Medien hört man immer mehr über CRISPR. Doch was ist eigentlich dieses CRISPR und was kann man damit anfangen? Um das herauszufinden, besuchen wir heute Iryna, eine Wissenschaftlerin aus dem IMB auf dem Universitätscampus in Mainz. Kommt mit! Hi.
Oh, hi.
Can you tell us, what is CRISPR and where does it come from?
I can tell you what it is, and the best would be if I show you some drawings.
The word CRISPR is an acronym, and it stands for Clustered Regularly Dispatched Short Palindromic Repeats. But nowadays, CRISPR is a more common name for a well-known gene editing technique. And the history of discovery of this technique goes back to the studies of bacteria and archaea. And the way it happens in the lab is the following. We provide nucleases, we provide RNA to specifically target nucleases to the site of the cut. We also give the complementary sequence to the for the DNA to be replaced. We incubate it together for some time, and then we add it to cells. Then when these complexes get into the cells, the guide RNA will guide the complex to the genomic position that we chose. And the DNA will be cut with Cas9. And the piece of DNA that we provided as a template will get inserted in the genome of the cell. Cells will then repair the DNA breaks that were made by Cas9, and in a few days we can start screening for CRISPR-Cas9 genome-modified cells. And now we'll show you how it's done in the lab. So in our lab, we're interested in finding out how genes and how proteins are regulated. And for this, we want to trace them. We want to see where in the cell they are in the nucleus, where they go in the cytoplasm, and we want to see if they're still there, if they're gone. And in the end, we want to find out general mechanisms of how proteins and genes are regulated. And for this, as an example, I'll show you how I'm creating tagged proteins. So I want to see where they are localized in the cell, and therefore we're using the CRISPR technique. I'll attach fluorescent protein to them that will show localization. And as I explained before, you have to prepare three things. So first you have to compose your gRNA, your guide RNA, and you have to know where exactly in the genome you want to make an insertion and where the Cas9 will be cutting. And there are special programs that help you to do this. All you have to know is where exactly you want to make the cut. Second thing, you have to develop a target DNA. So basically what you want to insert. In our case, that's a fluorescent protein, amnion green. You have to develop a plasmid which will encode this gene, and then you can, using the gRNA, put it together, using Cas9, make a cut, and it will get integrated in the genome. And the third component, your cells. And now I will show you how we do these things together. Because we work with cells, we have to be working very carefully without contaminating them. Therefore, we're using a laminar hood. So the air comes from that side and it's filtered. What we are doing is, I have already prepared samples with our guide RNA, the template, and a Cas9 source, and we will tag genes that have cytoplasmic and nucleus localization. So DNA cannot just go into the cell as it is, it has to be coated with a special reagent, with a transfection reagent. So I take a bit of it and add to the DNA sequence, to the DNA mix. Now DNA will be coated with the small particles and will be possible to enter cells and get integrated. We mix them together and we leave them to incubate for 10 minutes. So 10 minutes have passed, the complexes are formed, and now we can add them to cells. swirl the liquid nicely so that every cell that is attached to the bottom gets its share of DNA. And same with the other one. Now we'll leave the biology to do magic and put cells in the incubator where they feel warm and well cared of for a few days, and then we see the results. In the lab, we work with cancer cells because they can live forever and you can make nice experiments with them. Here you can see HCT116 cells that are derived from colon cancer cell line. And this is how they look under the microscope. Now we've seen that we transfected them with constructs for CRISPR-Cas9 technique. And now we can see that the protein that we are interested in is localized very nicely in the nucleus. So this is what you see here. Nice bright green spot means that all the proteins that we are interested in is tagged with the fluorescent protein amnion green and is strictly localized in the nucleus. And every cell now is having the tagged protein. Now remember we made two different constructs. Another gene is present in the cytoplasm as well as in the nucleus. And that's what we're looking for here. Again, healthy cells looking wonderfully happy. And you can see how the protein is now labeled, the one that we're interested in. It's localized in the cytoplasm and a bit less in the nucleus. And using CRISPR-Cas9 technique, now we're able to study behavior of these proteins and the control of their expression.
Wow, sounds so easy. Are there any difficulties?
Well, we wish it was so easy. It does sound easy, and it is an easy technique once you know what you're doing. The most tricky part is to select the right cell type that you want to study. Then you have to be really sure about the sequence that you want to bring in and to design properly your constructs. But even then, when things are well designed and well performed, there is still a mystery of biology behind. And that's why we do discoveries, and that's why we do research. We want to change gene functions, but we don't know yet what the outcome of these changes will be. So the most challenging part is afterwards to interpret the results. And that's what we are working on.
Thank you for showing us the work in the lab.
You're very welcome. And if you're passionate and curious about biology, you're very welcome to come and join us in the lab.